Good morning from Florida and good afternoon in Europe. Good evening elsewhere around the world. I'm Jim Lane, and I'm editor and publisher of the Digest. And welcome to today's Digest webinar. We've been looking forward to this for some time. The scale-up journey of bio-based processes, real-world challenges, surprises, and successes. Scale-up has always been one of the defining challenges of the bioeconomy. Great ideas are born in the lab every day, but commercial reality has its own rules, its own surprises, and occasionally its own sense of humor. For years, companies often had to learn these lessons the hard way, one project at a time, great expense incurred. But increasingly, the industry is building up something extraordinarily valuable we'll be exploring today, shared operational knowledge. In other words, scale up is becoming learnable. And today we're going to hear firsthand perspectives from organizations deeply involved in that journey, from pilot infrastructure to industrial biotechnology deployment, and we'll be exploring not only the challenges, but the lessons and the patterns and the successes that are helping move the sector towards the end of its journey forward faster and to get us to scale. Joining us today, we have Hendrik Waegeman, who is the head of business operations at Biobased Europe pilot plant in Belgium. From the great country of France, we have David Thomaset, who's the Chief Manufacturing Officer of Micropep Technologies. And joining us from Denmark, we have Abigail Jang, who is the Director of R&D at 21st.bio. There's a lot to unpack, and we're delighted you could join us. Please note that you're going to you have chat tools at the lower right of your screen. Please do, first of all, introduce yourself. Let's get you into the mix when we get to our Q&A at towards the top of the hour. If we know who you are and a little bit about your background, it's going to make that a little bit more successful. Also use that for a little networking for yourself. And again, that's towards the top of the hour when we are doing our Q&A. And we are also recording this webinar, and we will have that recording available to you later, sometime mid-afternoon tomorrow. And we'll send out a note for that for those of you who are looking for the recording. So with that, Hendrik, I'm going to turn it over to you.
Good morning, everyone joining from the US. Good afternoon, people from Europe. Very delighted to be here today with two great other speakers, Abby and David. What I would like to do to get started is also to give a bit of a point of view perspective from Biobased Europe pilot plant, showing a bit how we look at scale up, the challenges we've observed, and maybe the lessons that we can learn from that. So for those who don't know Biobased Europe pilot plant, we are an open access pilot and demonstration facility. We're actually one of the largest pilot and demonstration facilities globally, with infrastructure ranging from lab scale over pilot scales to think about 30 liter, 150, 1500, 15,000, all the way to also to some manufacturing, some early stage manufacturing. So we have a 75,000 liter fermenter, which I will talk a little bit more about later on, where we can do first commercial batches at competitive pricing. So our slogan is turning grams into tons, because honestly, it's often already
Russia, China, Iran, North Korea. Yeah. China serves as the bank for a lot of.
There's somebody in the background, so maybe mute yourself if you can. So there's it's sometimes already challenging to produce a couple of grams. So very often when people come to us, they have developed a very nice fermentation process to produce their product of interest. But what you see is that what's often lacking is a good understanding of the downstream processing to purify the product of interest from that fermentation broth. And it's basically where we step in and help them to, in combination with let's say R&D services, to scale up their processes and get to those first tons of material to put into the marketplace. So just some key figures. We were founded in 2008, became operational in 2010. We've invested more than 80 million euro in a very lean manner. So I think for people who have visited already in the past know that I think if you would like to build the same type of infrastructure again, you will probably spend much more than the 80 million. We've We've done already more than 1,000 projects so far for more than 350 companies, like globally. So a lot of our customers, of course, obviously come from Europe, but for years we had 25 to 30% of our projects came from North America as well. And we also have quite a nice number of projects from Asia. We're currently with 178 people. That number changes a bit day to day, to be honest, as we are still hiring also today to increase our capacity. And we have different quality systems in place, ISO 9001, which is more a general quality system, but also FSSC 22000, which means that we can also produce food grade samples. And we're based in Belgium. For those who don't know where Belgium is, you can see it here on the map. Based in beautiful Ghent, which is a medieval town, so not the worst place to be at when you want to follow up on your trials during your scale-up journey. So what is successful scale-up for us? And of course, when you ask different people, you might get different answers, but at least in our organization, when you ask us what is successful scale-up, it means new products on the market on the one hand, either manufactured by us, by our customer, or by larger custom manufacturing organizations, but also investments into new biorefineries. And we're pretty proud to say that already for more than 500 million euro, there has been invested into new biorefineries based on results that were obtained at the Biobased Europe pilot plant. So why is a pilot infrastructure? Why is it so important? Basically, when you go from lab scale to commercial scale, typically the technological risk will decrease, yet at the same time, the capital requirements will increase drastically. And it's especially at that intersection of pilot and demonstration scale where both the technological risk is still high, but also the capital requirements are high. And this is a very difficult phase for investors to understand. And nowadays, also, but the consensus is it's very hard also to invest in infrastructure, in own infrastructure for companies and to do their own piloting. It's actually much more efficient to work with organizations like the Biobased Europe pilot plant to get this done. However, there's actually a second value of that. You have the first value of that to get from lab to demo, to prove that your process works at the demonstration scale. And later on, we'll hear some nice stories about that as well. But there's actually a second value of that when you want to get from demo scale to production scale. There are lots of barriers and more than we like, a little bit more barriers in Europe sometimes than in other parts of the world. In terms of regulation and access to feedstock and financing and so on, but also in production, we believe. Building a first-of-a-kind facility can be quite challenging, and sometimes you need to have the market volumes to do it to begin with, and you need to have the experience to do it. And we believe that sometimes it can be smarter to do, let's say, this first commercial production at CDMO level. And how we distinguish ourselves from other CDMOs in terms of production at 75 cube is that we really have a fully flexible DSP line. So we can do 75 cube production 1 week, be producing product A with a certain DSP, and then the week afterwards do a completely different type of process with our flexible DSP unit operations. And this is just a nice video where you can see when the fermenter was installed into the building. This was in 2023, and we've been using this unit since 2024. One of the first runs we've done also resulted in a press release, was a collaboration with Vivici, in which we scaled up their dairy protein production, BLG or beta-lactoglobulin, at scale, first run, and it was successful. And then later on, since 2024, we have used the 75 cube fermenter already for multiple processes, and David will tell more about the work we've done with microprep in that regard. Successful scale-up is much more than just infrastructure. It's especially the know-how that is crucial, and so that's why I highlighted this here as well. So I think what's really crucial in successful scale-up is that knowledge, that expertise to know how to scale. This is something that we have in-house with the people at the Biobased Europe pilot plant that we also combined with engineering know-how. So all the infrastructure that has been built at the Biobased Europe power plant has been built by our own engineering team. It's also combined with TEA, techno-economic assessment, so that you can really see the effect of certain decisions that you make during the scale-up journey and to see the effect on production costs. This can be spreadsheet based, it can be SuperPro based, but we have both tools also in-house. And on top of that, we also work with external consultants. So we have set up the Scale Up Accelerator Network, basically where we work with veterans in the field of industrial biotech, and where they can, together with us, help you in a review of your process flow diagram, your mass and energy balances, your raw materials and feedstocks that you're using, and potentially also the impact it has on your wastewater treatment plant, a check on your fermentation process robustness, advice on downstream processing unit operations. Again, also an evaluation of your techno-economical analysis and any advice possible on supply chain and partnership strategy. So there's a lot of USP challenges, to be honest, and a bit too much also to just capture here in this short presentation. But I think one of the most crucial aspects, definitely in scale up of a fermentation process, is oxygen transfer. And if you see what determines oxygen transfer, is of course the fermenter design, the way the height to width ratio, the impeller design, the baffle design, cooling design, and so on. And of course, the parameters that you can set yourself: pressure, aeration, and stirring speed. And there's a bit of a misunderstanding on how processes scale from lab to 75 cube or even beyond. And that's why I'm giving this as an example. This is a performance curve of our 75 cube fermenter in terms of gas dispersion. And what you see on the x-axis is the aeration rate in VVM, and on the y-axis you can see the stir speed in RPM. And so basically what you get in such a fermenter, and this is different from a lab scale fermenter, if you use a high aeration rate but a low stirring speed, you basically get the effect of flooding. So the gas is not properly dispersed, you get low mass transfer, you get oxygen depleted zones. What you're basically doing is you're pushing air or oxygen just through the fermenter without being dispersed. If you increase the stirring speed but still have a high aeration rate, you get loading. So you get to some extent gas dispersion around the impellers, but there's no gas recirculation, so still a suboptimal use of your air. When you find a good balance between aeration and stirring speed, typically a lower aeration rate and a higher stirring speed, you'll get full dispersion and you get optimal use of the oxygen that you try to bring into the system. Now, why is this important? I already mentioned that for people who are experienced in running,
I'll be down in a minute.
in running fermentations, they typically use the settings of 1 VVM and relatively low stirring speed because they want to avoid shear stress. But when you run under those conditions, shown as point A here, you get a very poor mass transfer, and you get lots of wasted energy actually. So it's actually much better to work at the lower aeration rate in such a fermenter, a higher stirring speed, point B here. This is really the sweet spot that you could find in this specific scenario, where you have an increased mass transfer. So you have a better oxygen transfer rate, you'll have higher yields, you'll have better performance, but it's also better for the hardware because you'll get less shaking on the equipment. And as you can see here with this calculation, you will drastically reduce your energy consumption. 46% less energy, which corresponds to an operational cost or reduction in your operational cost of 220,000 euro annually, assuming a 300-day operation. So these are kind of things we can also advise our customers on. On top of that, just some general strategies for success in USP or upstream processing fermentation is choosing the right scale. A lot of people want to go to the largest scale as quickly as they can, but of course you have to de-risk your technology. So it's important to start also small enough, yet at the same time find a consensus with having it large enough to have, if you need to have certain product coming out of it, that you want to do certain application tests, and obviously you need to have certain volumes. So it's together with our team, we will try to find a strategy to basically find a consensus between those two needs. Prepare. I think it's very important to be fully prepared on not only how the run would be done, on how or what equipment needs to be used. For example, special setups in terms of continuous fermentation, special feeds, or in situ product recovery, but also like in what could go wrong. This is something that we pay a lot of attention to. Is like what if scenarios? What if this happens? How should we react? Because otherwise, you don't want to make those decisions on the spot if they could have been uh, prevented. Contamination, it's fermentation. There's 1,000 things that can go wrong, and typically it's not really forgiving in the sense that if one thing goes wrong, it's typically and yeah, game over. This is a bit different in downstream because in downstream, typically if a problem occurs, you'll still have a way to solve the issue. In our case, I'll come back to that later on. But in downstream, we will always collect the different streams after separation. So it means like if you don't have a proper separation, you typically have the chance to try it again on another unit or change conditions and so on. In fermentation, when you make a mistake, it's game over. So it's important also when it's game over to do root cause analysis and to learn from those mistakes and to see if you can change that for future runs. Um, Infrastructure is super important. Proper bioreactors, reliable utilities, duplicate utilities, something we've done also to be prepared if something happens unexpectedly. Of course, training protocols and so on and so on. Um, Control and measurements, it's something we pay a lot of attention to, not only with the typical HPLC and GC type of analyses, but more and more also using smart sensors to do online follow-up of your feedstock, typically glucose, but also your end products. Some work, for example, we've done with the company Perceptra on online monitoring of compounds. And of course, there's a lot of data that will be generated during the pilot runs. It's important also to look deeply into to the effects that you might observe because there's a lot of information in the data, so it's important to properly analyze it and to learn from that. DSP a bit similarly, I would say, but I already mentioned that it's a little bit more forgiving. So if something happens unexpectedly, you typically still have a chance to recover, reprocess, solve the problem, etc. But here's just an overview of the difficult things that we typically observe. Here, very important is try to do, well, at least our advice, try to do as much as you can on bench scale. We have a fully equipped DSP lab where we basically have always the smallest version possible of the unit that we have or the units that we have at larger scale. So we try to with the lowest effort possible to test already as much as we can on small scale. Then when you go to pilot scale, it's also worthwhile to do parallel trials. So basically, for example, harvesting 1,000 liter of broth or 10,000 liter of broth and really testing different downstream unit operation protocols. One that is maybe already validated so that you're sure that you have enough product, but you could keep some material aside that you could test then for maybe an improved version of your DSP. Written preparation is key. Again, similar to fermentation, having everybody on the same page in combination with what if scenarios. What if we don't see proper separation? Shall we go to, for example, a filter to a centrifuge? You need to be prepared for that you don't have to decide it on the spot. Although I think industrial biotechnology is a much safer industry than, for example, the typical chemical industry, it doesn't mean that we're free from potential hazards. There's safety concerns in what we do in terms of solvents that we use and products that we're producing, in pressures that we apply. So it's very important to take this into account. Of course, executing, again, making sure that you have enough data sets from all the runs. Piloting is expensive, so you want to get the best out of it. And if your product is out of spec, go into detective mode. Also try to figure out what the cause was. So with this, I hope I've been able to give you a bit of a general introduction on what are the typical scale-up challenges that we observe as a pilot facility, and I hand over the mic back to Jim to introduce the other speakers and go more into the specific cases.
Well, Hendrik, thank you very much for an excellent opening. We got lots to chew on. I want to encourage everyone to get your questions in. We'll be getting to those top of the hour. Lots coming in already, and more to come. So we're looking forward to having that. Our next speaker and presenter, David Thomasy, is the Chief Manufacturing Officer of Micropec Technologies, and we are ready to go.
Good morning, good afternoon, everyone. Very happy to participate to that webinar. First of all, I would like to thank Jim for hosting, and also thanks Biobased for the coordination and the opportunity given to share our bioprocess scale-up journey at Micropep. So, who is Micropep? Very quickly presenting the company. The company has been founded in 2016 in Toulouse, France, and is developing natural crop protection solutions based on the incredible potential of micropeptides. The company has raised 60 million since 2018 from prominent European and US investors, and has started its expansion in the US in 2022. At Micropep, we are on a mission to help building more sustainable agriculture for future generations through delivering affordable, reliable, and sustainable micropeptide-based crop protection solutions. So where do our active ingredients position in terms of size between chemicals and biologicals? So as you can see, they very much stand within small molecules with 5 to 25 amino acid long peptide. Our first bioprocess to be developed and upscaled required our first product to be commercially launched next year in 2027, a biofungicide with broad spectrum antimicrobial activity. The bioprocess has leveraged some key physicochemical properties, such like short, linear, and water-soluble peptide. I will very quickly, so you better understand the next slides, introduce you with our process. Our bioprocess is based on inclusion body technology to maximize peptide protection from biological degradation. It uses E. coli, fed batch fermentation, and a 6-step DSP to harvest the inclusion body, wash most of the cell impurities, ensure peptide cleavage and separation from the inclusion body, and finally concentrate and dry. And all that displaying high peptide recovery and purity. So let me describe now our scale-up and industrialization strategy that we could describe around 4 main pillars. The first one is building very early a manufacturing platform. For Micropep, it was up to 8 years before commercial production with key industry capabilities. Second, integrating early on industrial constraint consideration in the process design and development. So the list can be numerous, but a few examples are around raw materials, for instance, with managing supply upfront on some complex materials like yeast extract, enabling lot-to-lot variability monitoring, or making sure the process is compatible with the use of industrial sugar syrup, for instance. Another example is around applying oxygen uptake rate and temperature condition manageable at large scale. Selecting standard DSP technologies is another good example. Then 3, partnership and applying a multi-scale approach with, as a first step, establishment of local and worldwide recognized bioprocess development and scale-up network, considering that all cannot be done only internally. And on the second step, significant investment in building strong and long-term partnership with one key partner. So in our case, with Biobased Europe pilot plant, with a step-by-step scale-up approach, as Hendrik was saying just before. So starting with 100-liter scale, and then step-by-step moving to 1,000, 10,000, and finally 50,000 liter. And this with a strong field presence to increase synergy and educate the partner with our technology. And last but not least, de-risking at large industrial scale to generate real-life bioprocess data at 75 cubes, paving the way to successful industrialization. So I will now share, even if the end of the road was successful for us, obviously we have encountered a few challenges during our journey. One first example is around technical challenges, especially around downstream technologies that are not always easy to scale down. For us, it has mainly been around centrifugation, and I will present that example just after, but spread drying, for instance, has also shown discrepancy, but with a lower impact. So centrifugation. Centrifugation is used in step 2 of our DSP to ensure major cell impurities removal during the inclusion body washing. Lab scale down is nearly impossible for that technology. And during pilot scale-up runs, we have not well anticipated some specific behaviors at larger scale. As a consequence, during our first 5-batch run that we have done at 15 cubes scale, we have encountered product cooking, as you can see on the picture, in the bowl, leading to precipitates clogging the centrifuge, with direct impact on the peptide recovery, as you can see on the graph at the bottom on the left, with a very low peptide recovery. The troubleshooting has shown first that we were accumulating for too long product in the bowl. The product inside the machine was not cooled down enough. A pH adjustment that we have implemented during development to increase the packing in the machine has shown to be not necessary at this scale. And also a fourth lesson was to use an equipment slightly undersized compared to the volume to be treated, leading to longer operation time. So as you can see, still on the same graph, the corrective actions implemented on the successive batches have led to global performance back to expectation, with recovery between 70 and 80% on that specific example. Beyond technical challenges, we have faced other hurdles. One has been during the de-risking batch at 75 cubes, with longer operation time and impact seen on process intermediate quality and stability, leading to global recovery and formulability issues. Another one has been on the analytical method transfer. Sometimes we could think of that topic as being secondary when you are managing a tech transfer, but when your process monitoring needs precise live measurements, putting enough focus on this transfer becomes critical. On the success part, so years of preparation, careful planning and strategic decision-making led to successful bioprocess scale-up to large industrial scale, paving the way to commercial production. So that can be seen on three achievements to support that success. So first, on the robustness of our technology. So you can see on these graphs some results from 5 batch run at the same scale and exactly as applying the same recipe. And so on biomass and protein production, reproducibility is appears to be very good. Also on one specific purification parameter on the downstream, we actually see after peptide cleavage, a secondary form of the peptide losing one amino acid while staying active that is appearing. And we need to keep that secondary form as low as possible. And you can see also on the graph that the percentage has been kept very low for all the 5 batch. And another parameter also showing that good robustness, it's around storage stability, either at +4 or at ambient temperature. And after 12 and 24 months, with all the data beyond 90% of peptide stability. So this confirms benefits of integrating early on industrial constraint and working hand in hand with the scale-up partner. Last achievement I wanted to share with you is the scalability of the technology at 75 cubes. And so you can see also on the same parameters, the good scalability moving from 1 liter to 1,000 liter, 10,000 and 50,000 liter on biomass and protein production, as well on the peptide recovery during purification, with data very much alike. And now moving to my last slide. Micropep is among the first companies to cost efficiently bioproduce small peptides on an industrial scale, but this did not happen overnight. So I would like to reemphasize that it is never too early to start your scale-up journey. It is key to bring technical and industry expertise and diversity in the team you are building. Never make the economy of generating sufficient quality data at lab scale, but also real-life data at industrial scale. And always remember that it takes a village to successfully bring your bioprocess to industrial scale. Thanks for your attention, and I'm giving the mic back to Jim.
Well, thank you, David, for the continuation as we dive down into the world of companies, and you can see the scale-up journey that actually happens, and a happy ending, it looks like, for Micropep. But we've got some questions coming in. Those of you that would like to get your questions in before we get to the top of the hour, we'll be able to directly address your concerns. But now we're going to dive into the world of a little bit more of precision fermentation, maybe a little bit about the world of dairy proteins. We have Abigail Zhang is the director of R&D at 21st.bio, 21st.bio.
Thank you to Jim and for BBE for inviting me to speak. And I just wanted to talk a little bit about our journey and maybe stories from the front lines. Let's see. Okay, 21st Bio, who are we? We are a Danish American company. I hope you can tell I am not Danish. I actually sit in the labs in California. Our headquarters is in Copenhagen, Denmark, in which we have R&D, the executive team, and all your business functions sit in Copenhagen. In Davis, we are strictly R&D. And what do? We essentially produce proteins and peptides. Our technology is actually licensed from a company called Novonesis, which is a global leader in enzymes and microbes. And what they have done is they've developed technology, strain technology, that they have really, developed over the past 40 years that are made for scale up and made for production of their own enzymes. And they are robust, they're high expressors of protein, and they can withstand the rigors of fermentation at a production scale. So what we do is we use this technology to develop the production of proteins and peptides for food and materials and agriculture products for company around the world. And while we do help produce proteins for a lot of different companies, I've kind of focused this talk about one specific protein. And that protein is BLG essential flex. It's one of the products. So BLG is a dairy protein. It is a whey protein. And a little bit about this program, because I want you to understand how this is all working. We have two different kind of business models in our company. We have what we call projects, in which companies come to us with their protein in mind. It is specific, it is proprietary to them, and we have a custom development program for them. And this BLG is a little bit different. It is what we call our program development. So we essentially take on the R&D ourselves, and we develop the production from strain engineering to scale up for our partners. And partners can kind of essentially join our program, and we help them scale up or produce either in a CMO or in their own facilities. And again, this talk is to kind of take all those learnings and bring them into one. But essentially, our BL journey has been actually pretty fast. From the first days of strain construction to a self-GRAS was 2 years. And then we got a no questions letter asked from the FDA in about 2.5 years. So it's been a very quick journey. And I think one of the, benefits from having production strains that are ready to go is that you can essentially reach that commercialization platform in a very quick time. The host that we use for production is Aspergillus oryzae, which is a filamentous fungi. It is actually the first example of a mycotoxin-free Aspergillus oryzae for the production of recombinant novel food protein. So our BLD protein, it is bovine, so it is from a cow. It is a slightly different variant, so it actually delivers 45% more leucine, making it a little bit more nutritious than I think your standard variant. But it still has the same benefits of bioreactivity and has a very neutral taste, and then can thus be used in all these different applications, whether it be a hybrid dairy product or sports nutrition, clear drinks, weight management, a lot of nutrition-based applications. So as a background of a strain engineer, I will say that really our scale-up journey always starts with the strain. And I think in the perfect world, which we don't live in a perfect world, we like to see this linear path of strain engineering a protein to your lab scale fermentation and then to your analytical chemistry and DSP. But in reality, it is a feedback loop in which we are always learning, always testing. There's a lot of iterations of each thing to make sure that we get the good performance of our protein, that we have the right characteristics. Can it withstand the stress of fermentation? So we keep going back and forth until we really find the right balance in which you're getting the right combination of expression elements, optimizing the media and process conditions, and making sure that your protein is still characterized correctly, ensuring the right DSP process, and it's meeting the quality and purity requirements that is needed for your application. And I will say, this could go on forever, right? Perfecting perfecting your strain. But as we look into the process of scaling, I feel like we're going to echo a lot of the same sentiments in that don't skip this step. Don't skip this stepwise smaller to larger to smaller to even bigger. You can't go from a 30 liter to like a 50 cubic meter. Don't make that big leap of faith. What we do is a lot of work is done in-house before we even think about a larger scale. So at least for us at 21st Bio, we have 250 mil ambers, 30 liter tanks, a 300 liter and a 1200 liter tanks in-house. So this helps us test the process at various scales and help us go up and down to ensure that we have the right fermentation conditions to transfer to a production scale. So we go back and forth and test all the things. We try to stress things. We try to make things easy. We try to mimic scale as much as possible to make the transition smooth. And I think I'll touch that a little bit on the next slide. But this at lab scale and maybe small pilot scale is where we really try to learn the most about the strain. And I think it's important to learn what you can do and what you cannot do and test all sorts of conditions so you really understand how and when to push your strain and how robust it is. And really, once you get into the CMO, and there is a need to troubleshoot or adapt, having gone through this process will help educate those decisions moving forward about like, what if this and what if that? And I think, I'm kind of echoing what the speaker said before me. What we have really learned about the scale up journey is that the conversations with your CMO needs to start early as possible. And maybe that is one of the biggest learnings that we have. Start the conversation very, very early. What are the CMO's equipment and capabilities, both for USP and DSP? We scale at facilities in Europe, in Central America, and the US, and each facility is slightly different. Do they have the right impellers, equipment, and programming abilities for your process? How much power do they have for possibly viscous fermentations? What DSP equipment is on site? And how quickly can that fermentation be processed? We had an instance early on in our journey in which the particular CMO had very limited capabilities in their programmings. And they were trying to be very accommodating, in which the operator will actually sleep at the tank and maybe turn the dials as needed throughout the night. As much as we appreciate the accommodation, we really had to kind of pivot. And again, understanding what can and cannot be done on the lab scale really benefited at that moment. But also understanding what raw materials are available in that region of the world. Different sugars could be cheaper in Central America versus Europe. If your raw material isn't available, can it be easily sourced or can it be shipped in? This can not only affect the performance of your fermentation, but also your economics. Understanding how the CMO operates as far as security, sterility measures. Are there power outages? We have had power outage situations. And for how long? Is your strain and process going to be robust and flexible enough to survive these small outages? Another thing that we also know is kind of scheduling. We talked about how quickly can your fermentation be processed once it's finished into the DSP. Is your protein stable enough for that long at room temperature or at the fermentation temperature? It's stable for 6 hours, 12 hours, 24 hours. I think it's important to get all those little details right with your CMO. And I think kind of getting an understanding of all these things could lead you to how you might alter your small scale fermentations to mimic the scale up conditions or details in your tech transfer documents. And as far as challenges, I think a lot of us probably see the same challenges. Tech transfer, I will say, as detailed as we would like to be in tech transfer, language can be interpreted in many different ways, whether it be due to differences in cultures or interpretations of the process. We've encountered, is it an engineer or is it a molecular biologist or scientist? Even those people do not speak the same language or terms sometimes. It should be as very detailed as possible, and then the tech transfer should be altered according to the equipment and the capabilities of the facility. We always recommend a detailed walk through the process on both sides to ensure that both sides understand. But I think we all know from real life that maybe the same recipe for a meal could be given to two different people and you get very different results. That's why we always kind of recommend one of our team members to be at the site in the facility for the first couple of fermentations to kind of answer those questions that might not be as explicit in writing, and something like, does the seed look right? Is it supposed to be this viscous? As much as we would like to detail, there's nothing like seeing it firsthand to kind of answer those questions. But then they're also there as support for the team to any troubleshooting and answer any questions that you will only run into as you're going through the process. One other challenge is that we have found is experience with fungal fermentations. I think a lot of CMOs are very well versed in maybe E. coli or yeast, but not a lot of fungal fermentations. They are tricky, which then we always get the question, then why do you use them? Because you can make a lot of protein. You can make a significant amount of protein in fungal fermentations. But there is less experience with them, and they are tricky because they are longer fermentations. So this could then lead to contamination issues. It is a filamentous fungi, so the biomass is actually quite large and then can lead to viscosity issues. And this is kind of touching back to understand the capabilities of CMO and if they have the right power and the right impellers to run the fermentation. And I think maybe to speak on the whole of precision fermentation, CMO capacity. It's difficult to find capacity for your protein with the right equipment and the right needs, especially if you're looking for a food grade facility. Overall, the capacity of fermentation is low. I will say it's much better in Europe than in the US. But if we are in the business of producing sustainable proteins, it's really hard. And I feel like all the companies are kind of fighting for the same space. So if you're a company that's trying to upscale the process and produce samples for clients or customers or batches for FDA or EFSA approval, it can be frustrating to get the space when everyone is kind of rushing to get to market and there's maybe only a handful of locations really in the world that is available to you. And that's all I have.
Well, Abby, thank you so much for sharing the challenges that we could stop to share that's great.
Yeah.
So we're going to get into the Q&A now, and we have some questions coming in from our participants around the world, and thank you for those that are coming in.
Oh.
And let me just take some of those. I'll try to organize them a little bit, and I have a couple of my own that I want to get in before the hour is up. But we've got about 10 minutes or 9 minutes before we have to wrap, so we have a little bit of time. And so if you have a question that's burning, please do get that in. In the meantime, Hendrik, just a quickie that I think was lost in translation there. We had a question about what was the size of your largest fermenters. What's the largest scale that you go to at?
Yeah, so the total volume is 75,000 liter. That's yeah, The total volume, maximum working volume depends a bit on what microbe you're using. Also, is it the first one or the second or the third? Because of course, in the first one, you're going to be a bit cautious on not filling the fermenter to the top. So you could go working volume up to 55, 60,000 liter, depending on the process. So that then aligns with what David said on the 50,000 liter scale. He was referring to the working volume, where the 75,000, when I talk about it's the total volume.
Follow-up question on we had a fair amount of discussion there, especially in Abby's presentation about CMOs and handing off effectively your great process to a great CMO partner. Hendrik, do you have any? I mean, how do you handle that? How do you assist your members, your partners, in once they are scaled up to pilot in your hands and with your equipment? How do they start to access and how do they do their research on finding that really good CMO partner? There being never enough options, but they're not all the same. How do you help them through that part of their journey?
Yeah. So it's case by case. Sometimes companies have already kind of determined who they want to work with after they've done the scale up with us. So it's already a bit decided. That's one option. Um, a second option is that we really help them in also finding the most, fitting CMO, because it's like Abigail said, I mean, every project is also a bit different. The DSP is different. Timing is also different in terms of like when do you ask the CMO? Capacity might be an issue, but it also changes over time. There's years or months that they're tend to not be busy, and then suddenly they get all those requests in at the same time. So I think the advice we try to give to begin with, the advice we try to give to our customers is be as flexible as you can. So that's why when we do DSP optimization, we will typically have different versions of the process. You might be able to use a centrifuge, you might be able to use a microfilter, you might be able to use a filter press. So we keep it a bit open so that your process fits multiple CMOs. And then secondly, we help them navigate. There's customers that we basically toured around Europe to together with them find the most suitable CMO. So it's we also can do that as a service, right? And we know, of course, what are the pros and cons to every CMO?
Abby, can I just follow up with you just on that same question? Clearly, as you mentioned, there are CMOs that are right for different types of processes, molecules. It's never a right or wrong, but how do you help your partners and to think through that part of the journey?
Yeah, I think there are a lot of questions to answer, and I think the questions, I'm going to say cost is a big factor. Different CMOs are cost differently, and what is within their budget. And kind of answering the questions as far as like scheduling, maybe something in Central America might be a little bit cheaper than in Europe, and kind of answering those questions. But we do have cost, I think, raw material availability, scheduling availability, and what are the goals and timelines that is needed, for the partners. I think those are the questions that we ask first. And there is a little bit of a geographical question. Quite frankly, if you're going to produce a food protein, a lot of people go to the US first because the FDA process is a lot easier than an EFSA process. And so maybe they would like to have something in North America or Central America. It really depends on the goals of the partner.
David, a version of the same question to you. Where are you actually planning to do your production? Are you going to build, own, and operate, or are you going to work with a CMO for the long term, short term? What plans do you have for that part of your journey?
Yeah, thanks, Jim. Yeah, that's a very good question. So the approach today that we have, let's say short and midterm, it's to actually rely on a CMO. And we are in the process of qualifying a first one, but the goal is also to be as close as possible from the future market, and have also contingency plans. So probably also looking to have CMO at 2 geographies, probably one in Europe and one in America. And then for the long term, it will depend how the business at Micropape will develop.
Terrific. Let's in the few minutes that we have remaining, I wanted to get one of my own in here, so I'm gonna just steal the microphone here. Often when we do these webinars, we think in cautionary terms, challenges, and I wanted to ask Hendrik, I wanted to ask sort of on a more positive basis, what aspects of scale up, as you've seen your partners going through your system, have turned out to be easier, faster, or more manageable than expected? What goes right every once in a while?
Simple processes. I think that the more robust the process is from the start, the more straightforward the DSP is, the easier the scale up is. And I think that that's quite an easy one, of course, to say. But I think the we fast tracked some scale up programs primarily because it had been done before for other types of products, but using the same types of processes, or because the processes are robust and simple. But of course, that's not always possible. If you make certain new molecules that have never been produced before, you need sophisticated downstreams, and that requires, a lot of testing, a lot of different steps in the middle to be done so that you really de-risk until you go to 75 cube. But there are some processes where you can basically skip a lot of steps because they're very simple.
I'll take a volunteer, Abby or David, if you want to address the same thing. What goes right, that we can point to that maybe a surprise along the way that in your scale up journey that turned out to be easier than you had anticipated. David, maybe let's start with you, and then Abby, because you work with lots of different clientele on that. But David, maybe a thing or two.
Yeah. If I am looking at our experience with our first product, definitely fermentation has been the most pleasant surprise to us. And with some times of reflection on it, what I could say is that it's probably also the part of the process where we have generated most of the data at lab scale. I think it's also we're having the equipment. As long as we are integrating some industrial constraint early enough in the process development, I think when you do fermentation at bench scale, you could mimic a lot of the parameters that Hendrik and Abby was mentioning before. So definitely, we have been very amazed by the robustness of that fermentation reproducibility moving up at each scale. And I think it's due to that quality and quantity of data generated upfront.
Abby, over to you. A lot of work that you've done, a lot of different partners, so different ways to look at the journey. Any things that stand out that you had something that turned out a little bit more easier, faster, more manageable than expected, in addition to all the challenges?
I think maybe my answer is not a technical one, but maybe on relationships with the partner. I think the one thing that we sometimes fail to see is that everyone wants to succeed. Both parties want to succeed. And going into that relationship with knowing that, everyone wants to do well. And being able to work together and solve the challenges together with, your company and your CMO. We have been happy with the collaboration in troubleshooting and finding out solutions and answering questions, and working together.
Terrific. We got onto the human side of the scale up journey as well as the technical. We have time for just one last question. I'm going to give it to Hendrik. Two things that people, as they're beginning their scale up journey, first thing they're going to be doing, of course, to prioritize is calling you and forming a partnership and doing it right. What's the second thing that you would advise a brand new company calling you out of the blue tomorrow? was on this webinar and we're beginning our scale up journey, what would you advise for them to prioritize in those first 12 to 18 months?
Very difficult question, to be honest. Um, I think definitely think where you're heading towards. I think that's the first. A lot of people come to us without really knowing what their final goal is. Um, will they eventually build their own facility? Will they work with CMOs? How are they going to face basically their market introduction? How are they going to do regulatory approval? So I think building that strategy first before you come to us can only help to get the expectations right and to also allow us to think along with them. Because of course, when your ultimate goal is to build your own plant and be the cheapest as possible, the development will be different than when you say we want to match our process with a CMO, where you want to have flexibility, like I just said, that you're not dependent on one party, that you don't have to do major investments at the CMO. So it's knowing where you're heading at is super important.
So begin with the end in mind. Good advice
Exactly.
for those starting out. And of course, we have also need to be mindful of our end because we've come to the top of the hour. It's all that we have time for today. My great thanks to Hendrik Waegeman, David Thomasy, and Abigail Jang for joining us, sharing some experiences and insights from the scale up frontier. It's really important that you get these down. It's where exactly where you want to get those lessons learned from someone else's experience and not recreate the problems of the past at great expense. So thanks to each of you for joining us, and also each of you for joining us in the around the world for the webinar, making us a part of your day. Scale up may never become easy, but it is becoming more learnable and increasingly more repeatable. So I hope the discussion will help you accelerate your own journey. We do have a recorded version of the webinar ready for you shortly. Tomorrow, we'll also have a transcript that we'll share, and you can ingest that with your favorite AI and look into deeply into these comments and thoughts and learnings for some time to come. Until then, I'm Jim Lane, wishing you a great week ahead in the advanced bioeconomy and a safe one. So long. I'll see you next time.